Welcome back to the Deep Dive, where we slice through complex information to bring you the most compelling insights. Have you ever truly paused to appreciate the breathtaking complexity and adaptability of your own vision?
It's pretty incredible when you stop and think about it.
Right, think about it. You can transition seamlessly from like the blinding glare of direct sunlight to the subtle shadows of a dimly lit room.
Or track a tiny buzzing insect as it darts across your field of view.
Effortlessly.
Exactly.
Yeah.
Our biological eyes perform these miraculous feats without conscious effort, delivering this rich, continuous visual experience despite the incredible computational challenge happening beneath the surface.
A lot going on there.
So today, we're plunging into the cutting edge of artificial vision, specifically a groundbreaking development that aims to replicate this biological marvel, a TAW-cell-based analog silicon retina.
Yeah, this is really fascinating stuff. Our mission today isn't just to admire nature's engineering, though. It's to understand how brilliant minds are looking to the human eye itself, its elegant, event-driven processing, to build a new generation of vision systems.
Systems that are smarter, faster, and dramatically more efficient than anything currently available.
Right. This isn't just about taking better pictures. It's about processing the visual world in real time with an intelligence and adaptability that, mirrors our own.
Indeed, the motivation driving this research is truly profound. It's about fundamentally transforming how machines see by maximizing the transmission of relevant information and the detection of crucial features.
How do they do that?
Well, by incorporating essential biological principles directly into the hardware itself. We're talking about advancements that could revolutionize everything from prosthetic vision
Wow.
to the capabilities of autonomous vehicles and sophisticated robotics.
And for this journey, our primary guide is a meticulously detailed research paper. It's titled Tau Cell-Based Analog Silicon Retina with Spatiotemporal Filtering and Contrast Gain Control, authored by Prince Philip and his esteemed colleagues.
And it's important to stress this isn't just theoretical speculation.
No, definitely not.
It details the complete design, the precise implementation, and crucially, the rigorous testing of a physical silicon chip that actively mimics many of the most critical functions of our own retina.
So it's real hardware.
Real hardware. By the end of this deep dive, you'll not only grasp the inherent limitations that plague most of our current camera technologies,
Which we'll get into.
but you'll also understand why biology offers such a compelling blueprint for overcoming them. You'll discover how an artificial retina, built on these principles, could potentially restore highly nuanced vision for those with impairments.
That would be incredible.
And critically, how it could imbue robots and autonomous systems with a truly seeing capability moving beyond mere data capture to genuine intelligent perception.
Okay, I'm ready. Prepare for some genuine aha moments as we explore how these complex biological mechanisms are being translated circuit by circuit into silicon. So, let's kick off by confronting a fundamental question. Why are our standard digital cameras, the ones in our smartphones, security systems, even high-end film equipment, why are they still falling short when it comes to truly seeing and interpreting the world with the same natural efficiency as a biological eye?
Well, we're all incredibly familiar with frame-based image sensors, right? There's the ubiquitous technology powering pretty much everything.
Everywhere.
These sensors operate by capturing a series of discrete static images or frames one after another, like a flipbook animation, basically.
Okay.
And while they excel at capturing exquisite detail and producing high-resolution images in static or slowly changing scenes, their fundamental approach creates a significant bottleneck when the visual world gets, well, dynamic.
Dynamic meaning fast-moving or
Yeah.
complex.
Both, really. They often boast advantages like a small pixel pitch, which allows for high resolution, and a high fill factor, meaning a large percentage of each pixel's area is actually sensitive to light.
Which sounds good.
It is good for certain things. And engineers have also developed clever techniques like correlated double sampling, CDS, or delta reset sampling, DRS, to minimize noise, like fixed pattern noise, which can otherwise mess up the image quality.
So they're masters of the still picture, or maybe a smooth, slow pan. But where do they hit their limit? What happens when the visual information is constantly in flux, when you need to react to rapid changes?
Well, the ingenious aspect of these sensors often becomes their Achilles heel in dynamic environments. They capture every single pixel in every single frame.
Every pixel, even if nothing changed.
Exactly. Regardless of whether anything has actually changed in that part of the scene. Imagine trying to track a fly zipping across your living room with a camera that's simply taking, say, 30 static pictures per second.
Right, like a standard video camera.
You likely miss most of the fly's erratic path, or maybe just capture it as a blur. Meanwhile, the camera diligently records the static background, the wall, the furniture, over and over, frame after frame, even though it remains totally unchanged.
That sounds incredibly inefficient. All that data, and the vast majority of it, is just redundant information about things that aren't moving or changing. It's like constantly refreshing a web page that hasn't updated.
Precisely. This frame-based readout scheme leads directly to a massive waste of bandwidth. And if you want to capture faster motion and avoid blurring, what do?
Crank up the frame rate.
Right. But that isn't a free lunch. It has significant cascading consequences. You instantly generate a huge increase in power dissipation because more pixels are read out more frequently.
Makes sense. More work, more power.
You're also creating a massive amount of raw data that then needs to be transmitted and processed downstream. And here's the kicker. Much of that data is often, frankly, useless as it pertains to static parts of the scene.
So you're drowning in data you don't even need.
Pretty much. And if you then try to make these systems smarter by, say, detecting specific regions of interest within those frames, maybe looking for a face or a moving object that adds significant computational cost after all the redundant data has already been captured and transferred.
So you process the junk first, then try to find the good stuff.
It's akin to meticulously collecting an entire field of hay and then trying to find the needles, rather than having a system that only alerts you to the presence of needles in the first place.
Which highlights a critical point. Our biological retinas operate in a fundamentally distinct way. They don't capture frames, right? They're event-driven. Our eyes aren't taking 30 static pictures of your living room every second.
It's not at all. Looking at the larger landscape, the biological retina is just a masterclass in efficiency. Within our eyes, you have specialized photoreceptor cells, the rods and cones.
The light catcher.
Right. They don't constantly send a full pixel-by-pixel image. Instead, they primarily respond to changes in light intensity.
Only changes.
Well, significant changes. When a significant change occurs, a sudden brightening, dimming, or movement, they undergo an electrochemical process that generates nerve impulses, or spikes, which are then transmitted onwards towards the brain. That's the core idea of event-driven processing.
Okay.
Similarly, an artificial neuromorphic vision sensor, inspired by this biological principle, only sends data from pixels that detect a significant change in intensity beyond a predefined threshold. It only activates and sends information when something happens at that specific location.
So instead of that constant, undifferentiated stream of pixel data, the retina acts as a highly discerning gatekeeper. It's not sending a full image, but rather a compressed event-driven signal. Alert, this pixel just registered a significant increase in light. Or, attention, a sudden dimming here.
Exactly. It's a visual Morse code of change, you could say.
Yeah.
It dramatically cuts down the data while preserving the critical information about what's actually happening.
That sounds much smarter.
It is. This approach drastically reduces the transmission bandwidth because data is only sent from activated pixels. Think of it as a highly efficient news agency. It only reports breaking news, not a continuous stream of everything that's not happening.
And the benefits.
Several profound benefits flow from that. Incredibly fast response times because it's only reacting to changes. A wide dynamic range as it can adjust its sensitivity to events rather than absolute levels across the whole frame. Extremely low latency since processing begins immediately at the pixel level.
No waiting for the full frame.
Exactly. And highly effective in-sensor processing. The initial computational heavy lifting happens right where the light is detected, preventing that bottleneck of transmitting vast amounts of raw data to a central processor.
And this isn't a completely novel concept. I recall some early pioneers who recognized this elegance way back.
Indeed. The seed of this idea was planted decades ago.
Mm-hmm.
Pioneers like Carver Mead and Misha Mahald created some of the very first silicon retinas back in the late 1980s and early 90s.
Really? That or...
Oh, yes. They used things like diffuser grids to compute spatial contrast and included early bio-inspired components like photoreceptors, horizontal cells, and bipolar cells.
Yeah.
Their work really laid the foundational stone for what we now call neuromorphic engineering.
So people have been working on this for a while.
Absolutely. Others expanded on this, often focusing on individual aspects like spatial contrast, enhancing edges or temporal contrast for detecting motion. But typically, these earlier designs didn't combine the comprehensive suite of biological features that we're discussing today,
Like what?
like robust gain control mechanisms or the crucial distinction between different functional cell types. This new work by Prince Philip and his colleagues builds directly on that legacy, but it pushes for a far more complete and integrated emulation of the natural retina's multifaceted functionality. They're really striving for true bioplausibility in silicon.
Okay, so we've established the why traditional cameras are limited and why biology offers a potentially superior model. Now, let's truly dive into the core of what makes our eyes so incredible and how this new chip meticulously tries to mimic those biological secrets. The overarching goal here, as the researchers articulate it, is to maximize information transmission and feature detection by successfully incorporating a suite of essential biological features into hardware.
Right. It's about getting the most important visual information processed efficiently.
So what are some of those key biological superpowers they're targeting? Where do they start?
Well, for anyone familiar with visual neuroscience, the center surround receptive field is practically legendary. It's one of the retina's foundational superpowers, a brilliant biological trick for enhancing contrast, and it's absolutely critical for this chip.
Center surround. Okay, break that down.
Imagine having tiny spotlights on the visual world, but instead of just illuminating, they're constantly comparing a central point with its immediate surroundings. The response of a retinal neuron isn't just about the light hitting one spot, but how that spot compares to the light in a larger concentric area around it.
So it's not simply looking at a pixel in isolation, but how that pixel's light intensity dynamically relates to its neighbors. It sounds like a built-in difference detector.
Precisely. That's a great way to put it. This mechanism plays a vital role in promoting something called lateral inhibition.
Lateral inhibition.
Yeah, where the activation of one neuron tends to inhibit the activity of its neighbors. This dramatically improves our visual system's capacity to detect the edges of stimuli, the boundaries where one object ends and another begins.
Which is crucial for actually seeing objects as distinct things.
Absolutely crucial. If everything were uniformly bright or dim, it would be incredibly hard to tell where one thing ends and another begins. Lateral inhibition sharpens these boundaries, making them pop out.
Okay, that makes sense for edges.
And importantly, this isn't just a static comparison in space. It also has a crucial temporal dimension. A change in light in the center relative to the surround, or vice versa, is a very strong indicator that something is happening.
Like movement.
Exactly. Something is moving, or a new feature has appeared. It's a fundamental mechanism for motion detection and change perception, built right into the early stages of visual processing.
That makes profound sense. And our eyes don't just see edges. They also possess that incredible ability to adjust to light levels that vary by, 14 orders of magnitude?
Yeah, an astonishing range.
Ever walked from a dimly lit cinema into the dazzling afternoon sun or vice versa, your eyes adjust almost magically. That's luminance adaptation.
And this raises an important question. How does our visual system cope with light levels spanning that huge range? From picking out a faint star in a moonless sky to handling the blinding glare of a welding torch, all within seconds, and still discerning fine detail.
How do they do it?
Well, photoreceptors alone simply can't achieve this. They saturate pretty quickly at high light levels. So we have multiple stages of gain control operating in concert. Luminance adaptation is one such global mechanism. It involves physical changes like your iris constricting.
Right, the people getting smaller.
And complex biochemical adaptations within the photoreceptor cells themselves. But then there's a more localized, sophisticated mechanism called contrast gain control.
Okay, contrast gain control, how's that different?
This specifically happens a bit later in the processing chain, between the bipolar and amacrine cells in the inner retina. This mechanism dynamically adjusts the sensitivity to local contrast.
Local contrast, so not the overall brightness, but the differences nearby.
Exactly. It's about maintaining perceptual sensitivity in widely varying conditions. So whether you're looking at a subtly textured wall in dim light or a vibrant high-contrast pattern in bright sunlight, your eye adjusts its local sensitivity to ensure you can still perceive and distinguish those critical differences in contrast. It's like having an automatic exposure setting that is vastly more sophisticated and instantaneous than any camera could ever hope to be.
It's truly a marvel of biological engineering, and I understand our retinas have even more specialized detectives at work, different cell types with distinct response profiles.
Yes, absolutely. Beyond just adapting to light, our retinas contain specialized types of cells that respond in unique ways to visual stimuli. This differentiation is crucial for a comprehensive understanding of the visual world.
What kinds of cells are we talking about?
Well, for example, we have tonic or sustained cells. These cells have a remarkably low activation threshold and respond continuously, providing a sustained output as long as a stimulus is present.
So they signal, this is still here.
Pretty much. They are highly sensitive to low-contrast stimuli, allowing us to perceive subtle details and enduring features. In contrast, you have transient or phasic cells.
Transient or phasic?
These have a higher activation threshold and respond only briefly, often with a rapid burst of activity. Then they quickly adapt and return to a baseline.
What are they good for?
They're particularly sensitive to high contrast changes, like a sudden flash of light or a rapid movement. Emulating these specialized cell types is key for an artificial retina,
Why?
because it allows the system to detect both the continuous presence of subtle stimuli and extract important dynamic features from a rapidly changing visual field, essentially seeing both the forest and the trees, regardless of their visual prominence or speed.
Okay, so different cells for different jobs, and ultimately our brains don't receive a continuous high bandwidth video feed from our eyes, right? They get a highly processed, compressed output in the form of spikes.
That's exactly right. The biological retina's output is not an image, not like a digital photo. It's a spike train, a series of precisely timed electrical pulses.
Like Morse code again?
In a way, yes. This spike train contains highly compressed and incredibly efficient information about the scene. It encodes changes and features rather than raw pixel values. This deep dive aims to meticulously replicate that process.
So the goal isn't rendering a picture for a display.
No, the goal is generating meaningful events or messages, these spikes, that can be directly interpreted by a neuromorphic computing system, much like our own neurons communicate.
Oh.
It represents the ultimate translation from the complex analog signals of light into the, let's call it, digital language of the brain.
We've now charted the incredible capabilities of the biological retina, from its event-driven efficiency to its nuanced adaptation and specialized cell types. But the truly monumental challenge, the holy grail for engineers, is translating these exquisite biological principles, which are inherently analog and often quite messy, into the precise functional world of silicon. How did Prince Philip and his team bridge that chasm? What's the architectural blueprint that allows this artificial eye to mimic life itself?
Well, the foundation of this analog silicon retina isn't some speculative design cooked up from scratch. It's built upon an established software model of the retina called Convis.
Convis, okay.
This model is a sophisticated, validated computational framework that already includes all the major neuron types found in a biological retina, from the initial photoreceptors right through to the ganglion cells that send signals out to the brain.
That sounds like an incredibly smart approach. Starting with a proven, comprehensive model, it implies a deeper understanding than just picking isolated features.
Absolutely. The power of this approach is that it allows for a systematic and rigorous translation. You go from a well-understood, validated biological model directly to a hardware implementation.
Gives you more control.
Exactly. It gives the designers complete, principled control over their architectural choices, rather than just, piecing together disparate circuits hoping they'll work together.
Makes sense.
Furthermore, they made a critical decision early on to use a current mode design approach for implementing the various functions and filters.
Current mode. Why is that significant?
For those who delve into circuit design, this choice is particularly significant. Current mode circuits often make it easier to realize the complex, nonlinear equations and linear filters that are absolutely essential for mimicking biological processes.
Like what kind of processes?
Things involving multiplication, division, and other nonlinear dynamics that happen all the time in neurons. This approach is also often more robust to supply voltage variations and can be more energy efficient.
Which is crucial for something like...
Exactly, crucial for applications like retinal implants, where power constraints and precision are paramount.
Okay, let's unpack the very first major processing stage right after the light hits the photoreceptors. The outer plexiform layer, or OPL. This is where the initial crucial filtering and comparison really kicks off, acting like a sophisticated preprocessor for the raw visual data.
Indeed. In the OPL, the raw luminance input, that initial light signal detected by the photoreceptors, isn't just passed along passively, it's actively processed right away. This layer transforms that single luminance signal into two distinct and opposing signals: a center current, IC, and a surround current, IS.
Ah, the center and surround again.
Precisely. These two currents are then meticulously subtracted from each other, IC minus IS, to produce the final OPL output current, IOPL. This elegant subtraction is the fundamental essence of lateral inhibition we talked about.
So it starts enhancing edges right away.
Immediately, it begins the process of enhancing visual edges and local contrast from the get-go.
And I believe there's a key novelty in how they implement this center surround filtering that sets it apart. It's not just a standard spatial filter, is it?
That's right. What truly stands out here is their use of a non-separable center surround filter.
Non-separable, meaning?
Unlike many simpler filters that treat spatial information, where things are, and temporal information, how things change over time, as separate processing streams, this design inherently fuses them. It explicitly accounts for the natural delay of the surround signal transmission observed in real retinas.
There's a delay in the surround signal.
Yes, a tiny one, just a few milliseconds in mammals, yet it has major perceptual repercussions in how we interpret movement, because this filter is designed to handle this temporal asymmetry.
Okay.
The spatiotemporal OPL filter can detect both edges and movements simultaneously, even in areas with uniform brightness where a static image wouldn't show any feature at all.
Wait, both at the same time? How?
Because the timing difference between the center and surround signals is the movement cue. Think about it. If something moves across the receptive field, the center signal changes first, then the surround signal changes slightly later due to that biological delay. The non-separable filter is explicitly designed to pick up on this precise spatiotemporal pattern.
Ah, I see. So it's not just difference, but difference over time.
Exactly. This isn't just a clever engineering trick. It's a fundamental shift in processing. Imagine a self-driving car encountering a subtle visual cue, perhaps a slight ripple on a puddle indicating a car just drove through, or the barely perceptible shift of a pedestrian stepping onto the curb in low light.
Things a normal camera might miss or struggle with.
Right. A traditional frame-based camera might struggle, treating these as separate, ambiguous data points across frames. But with a spatiotemporal filter that inherently understands the dynamic interplay of space and time, the system can instantly recognize a moving edge as a single coherent event.
That dramatically reduces ambiguity.
And enhances real-time decision-making. It provides an intuitive understanding of the world that goes far beyond static pixel analysis.
That capability detecting edges and movements simultaneously sounds profoundly powerful. But as engineers know, every added layer of complexity often comes with trade-offs. What were some of the inherent challenges in implementing such a sophisticated non-separable filter in hardware? Maybe in terms of power, area, or even manufacturing tolerance?
That's an excellent point, and you're right. Implementing such a non-separable filter in a compact, low-power analog circuit is a significant challenge.
How so?
Well, the inherent delays in analog circuits can be susceptible to variations in manufacturing processes, those tiny differences between transistors. Also, supply voltage fluctuations and temperature changes can easily throw off the delicate balance needed for precise spatiotemporal dynamics.
But so keeping that timing right is tricky.
Very tricky. The cleverness lies in using robust current mode designs and carefully chosen circuit topologies like the TOS-L building blocks we're about to discuss that can maintain their timing characteristics despite these real-world imperfections.
TOS cells, what are those?
They are fundamental circuit building blocks used here. Think of them as tunable electronic time constants. Specifically, they implemented a temporal low-pass filter, E.
Okay, a low-pass filter smooths things out over time.
Exactly. It's built using a log domain circuit design and one of these TOS cells. Its function is to mimic synaptic delays and the integration of synaptic currents. Essentially, it blurs signals slightly over time, averaging them out.
And the benefit?
A key advantage highlighted is that its time constant is independent of the input signal. This means its temporal filtering characteristic doesn't change wildly just because the input brightness changes, which adds stability.
Okay, so that's the low pass. What else?
Then they build a temporal high-pass filter, T. This filter captures short-term luminescence adaptation. It's implemented cleverly by simply subtracting the output of the low-pass filter, E, from the original input signal.
So input minus smoothed input equals changes.
Precisely. It highlights the changes in luminance over short time scales. And here's where it gets really interesting in terms of those specialized cells we mentioned.
The tonic and phasic cells.
Yes. By simply changing the time constant associated with the high-pass filter, which they can do externally by adjusting a bias current, this artificial retina can replicate the behavior of both tonic, sustained, low threshold, sensitive to low contrast, and phasic, transient, high threshold, sensitive to high contrast cells.
Just by tuning that one filter.
Essentially, yes. Adjusting that time Constant changes the cell's sensitivity profile, allowing it to mimic either the sustained response of a tonic cell or the brief, change-detecting response of a phasic cell. This offers incredibly diverse visual processing capabilities from the same core circuit.
That's remarkable flexibility.
It really is.
Hmm.
It means the chip isn't locked into one mode of seeing. It can be adapted.
So with those Tossell filters, we've gained incredible control over how the retina processes information over time. But vision isn't just about temporal changes. It's also about interpreting patterns and textures across space. How does the silicon retina replicate the way our own eyes blur, integrate, and pool information from neighboring cells to build a coherent spatial understanding? That brings us to the spatial low-pass filters.
Exactly. Spatial filtering is crucial for tasks like blurring out noise, reducing fine detail to see larger shapes, and detecting broader patterns. It mimics several biological processes like the electrical coupling between neighboring photoreceptors,
Where they influence each other directly?
right, or the synaptic integration that occurs in cells like horizontal cells, which gather input from multiple photoreceptors. Mathematically, it's akin to performing a spatial averaging operation over a local neighborhood.
How do they implement that in silicon?
The researchers here employ a diffusion network to compute this spatial averaging. This is a common and elegant technique in neuromorphic engineering. Imagine signals diffusing outwards from each pixel, like ripples in a pond, and influencing their neighbors.
Okay.
What's particularly ingenious is how they implement this diffusion. Instead of traditional resistors, which can be bulky and difficult to integrate efficiently and tune in analog circuits, they use transistors operating in the subthreshold region to act like tunable resistors.
Subthreshold meaning very low current.
Exactly. Operating transistors in this ultra-low power regime allows them to act like very high-value controllable resistors. This allows the blurring effect, or diffusion length, to be finely tuned by simply adjusting a control voltage or current.
Ah, so you can control how far the blur spreads.
Precisely. And this connects directly to biology. For example, horizontal cells, which are part of this lateral inhibition network, biologically have much larger receptive fields than individual photoreceptors. They pool information over larger scales in the visual field.
Okay.
This corresponds to needing a larger sigma value, or greater diffusion length, in their spatial filter, allowing for broader contextual integration. Meanwhile, photoreceptors have smaller receptive fields for fine detail, corresponding to a smaller sigma. The diffusion network allows this tuning.
It's an intricate dance between temporal and spatial processing. Now, our visual world is incredibly diverse in brightness, spanning those 14 orders of magnitude you mentioned earlier. From a moonless night to a sunny beach, our eyes are constantly adjusting, and this chip is designed to emulate that same profound adaptability. This is where contrast gain control comes back into play, right?
Absolutely. The visual system must cope with this astronomical range of light levels. It's far beyond what any single photoreceptor can handle without saturating or becoming insensitive. This sophisticated contrast gain control mechanism, which biologically occurs primarily between bipolar and amacrine cells, is essential.
And it adjusts based on local contrast.
Right. It adaptively adjusts the output gain of the visual signal based on the local contrast in the scene. It's a nonlinear feedback loop that dynamically tunes the retina's sensitivity based on what's actually happening in that specific part of the visual field, not just the overall illumination.
How is that modeled mathematically?
The mathematical model for this is often described as a leaky integrator with shunting inhibition. It sounds complex, but the core idea is that the leak conductance of the integrator circuit effectively determines the gain of current integration.
Leaky integrator, like a bucket with a variable hole.
That's a decent analogy. The leakiness, the leak conductance, GA, effectively has a divisive effect on the signal. Essentially, the leakier the system, higher GA, the less sensitive it is to input, and vice versa.
That makes sense. Conceptually, more leakage means less signal buildup. But how is this leak conductance intelligently determined in the silicon retina? How does it know what the local contrast is to adjust the leak?
This is where it gets clever. The leak conductance, GA, depends on a spatiotemporal neighborhood of contrast magnitude. This relationship is characterized by a nonlinearity function, QI-BIP. To effectively implement this Q function, the researchers devised a truly ingenious, novel, absolute value circuit.
Absolute value circuit.
Yes, designed using current mode circuits. Think of it like a smart local contrast meter for each pixel. It measures the magnitude of change or difference, how different a pixel's signal, IBIP, is from zero, essentially representing its contrast activity, regardless of whether it's an on or off signal positive or negative change.
So it just cares about how much activity there is.
Exactly. It produces a full-wave rectified response, giving it a clear, positive signal representing how much visual activity or contrast is happening in that immediate area.
And how does that control the gain?
If that local activity or contrast is high, this absolute value circuit's output increases, which in turn increases the leak conductance GA of the main integrator. This effectively dials down the sensitivity of that pixel's pathway to avoid being overwhelmed by the intense stimuli.
And if the contrast is low?
Conversely, if the contrast is subtle, the absolute value circuit's output is low, the leakage decreases, lower GA, and this boosts the sensitivity to pick up those faint details. It's a remarkably elegant, self-regulating mechanism.
Wow.
It's a core part of how our eyes maintain clarity across vastly different lighting conditions.
Yeah.
The final gain-controlled center and surround currents are then carefully subtracted as before to form the final bipolar current, IBIP, which now has this adaptive gain baked in.
So it's constantly self-calibrating its sensitivity based on the richness of detail around it. That's a huge leap in mimicking biological robustness. Now, as we move towards the final stages, this is where the continuous analog signals begin their transformation into the discrete, meaningful spikes that our brains ultimately receive. This is the inner plexiform layer, IPL, and the ganglion cells.
Indeed. We're getting close to the output stage. Before reaching the ganglion cells, the bipolar cell current, IBIP, which has undergone all that sophisticated spatiotemporal filtering and gain control processing, is passed through one more stage. A static nonlinearity function, NIBIP.
Another function. What does this one do?
This function, cleverly implemented using simple diode-connected transistors, essentially acts as another gatekeeper. It chops off negative data, or more precisely, performs half-wave rectification.
Half-wave rectification only lets positive signals through.
Exactly. This isn't just an arbitrary filtering step. It beautifully mimics how biological synapses often transmit information. Synapses frequently have a threshold and only pass along excitatory signals, effectively filtering out weaker or inhibitory signals below a certain level.
So it cleans up the signal further.
It ensures that only relevant, sufficiently strong, positive signals are carried forward to the spiking stage, further compressing the information and reducing noise. It's like a final check before sending the message.
So it's filtering out irrelevant or inhibitory signals, only allowing positive, meaningful inputs to proceed, much like a neural gate.
Precisely. And then we get to the ganglion layer. Now remember, while non-spiking neurons do most of the complex analog calculations up to this point in the biological retina,
Right, the OPL and bipolar cells.
the ultimate output that leaves the retina and travels along the optic nerve to the brain is a spike train, those discrete electrical impulses. Therefore, ganglion cells are specifically responsible for generating these spikes.
The final output stage. How is that modeled here?
This deep dive utilizes an established and well-regarded model for neural spiking. Leaky integrate and fire, LIF, neurons, to mimic these ganglion cells. Specifically, they employ the Mihalis-Niebuhr neuron model, which is a particular variant of LIF known for its biological plausibility.
And how is that implemented?
Again, they leverage the advantages of log-domain current mode circuits for a compact and efficient implementation.
Okay, so how does that LI process work to generate the actual spikes from the continuous IBIP current? What's the mechanism that translates the analog strength into a digital event?
It's a classic neurocomputational model brought beautifully to silicon. The incoming excitatory current from the bipolar cells, let's call it IGANG, derived from IBIP after the nonlinearity, first flows into a first-order low-pass filter, essentially another carefully tuned TAW cell.
Another smoother.
Yes, but this filter models the leaky integration of current within a neuron's membrane. Imagine charge slowly building up on the neuron's membrane, but also leaking away slowly over time. This creates what's called a membrane current, or EMEM.
So IMEM represents the cell's internal excitement level.
Exactly. Crucially, this IMEM is constantly compared to a predefined threshold current, I. Think of I as the trigger point.
Okay.
When IMEM surpasses I, when the cell's integrated excitement level crosses that critical point, boom, a digital spike, represented by a voltage signal Veen spike, is instantly generated.
The fire part of leaky integrate and fire?
Precisely. This Veen spike signal then does two things. It triggers the creation of a brief, positive output voltage spike, V spike, which is the actual digital output signal.
The message sent onwards.
And it also activates a reset mechanism that quickly drains the charge from the integrator, resetting an IMM back below the threshold, ready to start integrating again. This is the precise, energy-efficient signal that would ultimately be sent to higher brain centers, containing the compressed and efficient information about the visual scene.
It's a beautiful and elegant translation from continuous light intensity and complex analog processing to discrete, information-rich neural events. This all sounds incredibly sophisticated, a true triumph of bio-inspired engineering. But it's not merely a theoretical exercise or a simulation, is it? You mentioned earlier the researchers actually fabricated a physical chip. They took this blueprint from concept to tangible reality.
That's a critical distinction, yes. The researchers didn't stop at simulations, which are essential but only part of the story. They actually fabricated a single pixel of this retina using a standard CMOS 0.18 process technology from TSMC.
Standard manufacturing process.
Yes, nothing exotic. A mature industrial-grade semiconductor process. This physical implementation is paramount because it unequivocally demonstrates the feasibility and practicality of building these complex bio-inspired circuits using modern techniques.
So it's not just a drawing board idea.
Definitely not. The single pixel circuit itself physically encapsulates all the intricate building blocks we've discussed, the OPL, bipolar, and ganglion cell layers, complete with the necessary connections to neighboring pixels for the spatial filtering. This isn't just a proof of concept. It's a tangible step towards real-world, scalable artificial vision systems.
So once you have this physical hardware, the next equally critical step is to rigorously validate that it actually sees and processes information in the way it was designed, and more importantly, in a way that truly mimics biology. How thorough was this validation process?
Oh, it was exceptionally thorough. They used a combination of both physiological and computational benchmarks, which is really the gold standard. They meticulously compared the analog hardware's responses not only to their own established software model, the Convis model,
Right, the one they based the design on.
but also to foundational established biological observations from real retinas. For instance, they looked at the impulse response of the center surround OPL filter.
What's that exactly?
It's essentially how quickly and strongly the OPL responds to a sudden, brief flash of light at the center of its receptive field. And they found that the response measured from their chip, strikingly, resembles the one quantified by Schnauf in the retina of the macaque monkey.
Wow, directly comparable to a primate retina.
Yes. This isn't a casual resemblance. It's presented as a direct, robust, and highly significant link to biological accuracy. It provides strong evidence that the chip is responding to basic changes in light in a demonstrably similar manner to a real primate retina. It's a powerful validation of their bioplausible design choices.
That's quite an achievement, matching specific physiological responses. But beyond a simple flash, how well does it track and interpret complex changes over time? I understand they use something called a chirp stimulus for temporal accuracy.
Indeed, simple flashes are good, but you need to test dynamic responses too. To rigorously test its temporal accuracy, its ability to dynamically respond to continuously changing visual stimuli, they employed a chirp stimulus.
A chirp, like a bird sound.
Sort of, but for light. Imagine a sound that not only changes pitch but also volume over time. A light chirp stimulus is conceptually similar. It might start with a simple pulse, then the intensity might gradually decrease, then increase, then decrease again, followed by a series of oscillations that progressively grow in both frequency and amplitude.
So it really pushes the system through its paces.
Exactly. This complex stimulus is specifically designed to probe a system's response across a wide range of temporal frequencies and intensity changes simultaneously. They meticulously use identical parameters for both the circuit simulation of the hardware and the established Convis software model.
And the results.
The results were remarkably consistent. They showed close matches between the analog hardware's responses and the software model across all key stages: the OPL output, the bipolar cell current, and the input current to the ganglion cell layer.
How close is close?
They quantified it using variance fractions, a statistical measure of how much the variation in the hardware's output could be explained by the software model's output. The numbers were incredibly high: 94.8% for OPL, 95.96% for bipolar, and 94.93% for ganglion current.
That sounds very high.
It is. This is a crucial finding. It indicates that the outputs are sufficiently similar that any differences become imperceptible when generating spikes. In essence, for all practical purposes of triggering spikes, its temporal behavior is indistinguishable from the validated software model.
And beyond dynamic changes, how well does it perceive static patterns in complex images? Did they only focus on temporal responses, or did they also assess spatial
Oh,
accuracy?
they absolutely assess spatial accuracy with equal rigor. It's no good just seeing motion if you can't see shapes.
Hmm.
To determine how well it processes patterns and details, they use two well-known visual benchmarks.
What were they?
First, a sinusoidal grating stimulus, which is a pattern of alternating light and dark bars of varying spatial frequencies. It's commonly used in vision research to test edge detection and spatial resolution. Second, they use the iconic Lena image,
Ah, Lena, the standard test
the
image.
very same. It's a standard test image in image processing known for its diverse textures, edges, and smooth regions, a good all-around challenge.
How did they compare the results?
They employed a metric called the Structural Similarity Index measure, SNAM, to objectively compare the output images generated by the physical hardware pixel simulations with those from the software model.
SM, what does that measure?
It measures the similarity between two images based on structure, luminance, and contrast, aiming to be closer to human perception than simple pixel difference. A value of 1 indicates perfect similarity.
And the scores.
They achieved a global SIM value of 0.93 for the grading and 0.91 for the Lena image. These are excellent scores, demonstrating very high spatial replication. It means the chip is effectively seeing and processing the patterns and details of an image in a way that incredibly closely mirrors the precision of the software model.
But real-world devices aren't built in a perfect vacuum. How does this intricate analog design hold up against the inevitable variations that occur in manufacturing processes, or subtle fluctuations in supply voltage, the wide range of operating temperatures it might encounter? This is crucial for real-world deployment, isn't it?
That's an extremely pertinent question, yes. Robustness is key. The researchers tackled it head-on with a thorough PVT analysis.
PVT.
Stands for process, voltage, and temperature. This comprehensive analysis examined the chip's performance under variations in supply voltage they tested from 1.6V to 2V, and across a broad temperature range, minus 40 degrees Celsius to plus 40 degrees Celsius.
That covers most conditions.
It covers most industrial and environmental conditions, yes. Crucially, they also tested it across different MOS transistor corners.
Corners. What are those?
These represent worst-case manufacturing variations for the transistors. Think typical TT is the average, then slow, SS, where transistors are slower than expected, fast, FF, where they're faster, and the mixed corners, SF, FS. Designers use these corners to ensure robustness against unavoidable manufacturing inconsistencies.
And how did the chip perform under these stresses?
Remarkably well, according to the paper. Even under these extreme and varied PVT conditions, the output images generated by the simulations exhibit visual similarities and, critically, edges are distinguishable. This indicates that the core functionality remains intact, even at the edges of its operating envelope.
So it's pretty tough.
It demonstrates a remarkably high degree of robustness, which is a significant engineering achievement for such a complex analog circuit.
And beyond the overall manufacturing process variations, what about subtle individual component imperfections? Like tiny differences between supposedly identical transistors on the same chip. How do they account for that inherent variability in analog design?
Mismatch, right.
Exactly. Mismatch is always a concern in analog. To verify robustness against these more granular process and mismatch variations, where identical transistors might behave slightly differently due to microscopic manufacturing tolerances, they conducted a Monte Carlo analysis.
Monte Carlo, like the casino.
Hey, named after it because it involves randomness. They performed 200 simulation runs, each time introducing small, random variations to the transistor parameters, mimicking the real-world manufacturing spread.
And the outcome.
This statistical technique simulates the effects of these random variations on the overall circuit performance. The results, often visualized as a shaded region around a mean response curve, consistently demonstrated acceptable variance. This confirms the design's statistical reliability and its ability to perform predictably despite these inherent analog imperfections. Okay, so it handles manufacturing variations. What about real-world visual noise itself? Like random light fluctuations hitting the sensor or inherent electrical noise generated within the circuit. How does this silicon retina clean up its own act, so to speak?
That's another excellent point. The beauty of this silicon retina's bio-inspired design is that it actively minimizes noise at multiple stages. It's not just passively susceptible.
How?
Firstly, the sophisticated filters implemented in the OPL stage Those spatiotemporal filters are inherently designed to minimize the noise produced by the circuits themselves. They perform averaging and differencing operations, which tend to cancel out random fluctuations. It acts as an initial line of defense.
Okay, filtering helps. What else?
Furthermore, that critical subtraction operation at the bipolar layer, where the center current, I plus BIP, and the surround current, IBIP, are combined to form the final IBIP current.
The lateral inhibition step.
Exactly. That differential processing provides another powerful noise reduction mechanism. If noise affects both the center and surround pathways similarly, subtracting them cancels out that common mode noise. This effectively reduces the noise that propagates through subsequent stages of the silicon retina.
Ah, clever. So noise that's present everywhere gets reduced.
Right. So it's not just robust to external conditions and manufacturing variations. It also has built-in multi-stage mechanisms to actively clean up its own signals.
Right.
This ensures that the spike train sent to the next stage is as clean and reliable as possible.
Which is paramount for practical applications.
Absolutely. You need clean, unambiguous visual data for reliable perception and decision-making.
This TOS cell-based analog silicon retina is clearly pushing the boundaries of what's possible in artificial vision. It sounds incredibly capable. What are some of its truly unique advantages compared to other approaches, and where could this groundbreaking technology take us in the future?
Well, one of the most incredibly powerful features, and a true differentiator for this specific design, is that this retina isn't a one-trick pony. It's profoundly reconfigurable.
Reconfigurable? How so?
This means it offers the unprecedented flexibility to externally adjust its time constants and spatial constants, simply by varying specific bias currents and voltages that control those TOS cells and diffusion networks we talked about.
So you can tune how it behaves after it's built.
Exactly. Why is this so incredibly important? Think about the astonishing diversity of vision in the animal kingdom. A cat retina, optimized for low light hunting and rapid motion detection, has very different characteristics, different time constants, different spatial receptive field sizes than a primate retina, which is tuned for high acuity, color vision in brighter light.
Okay.
This chip could, in theory, be programmed to emulate specific aspects of these different biological retinas. That makes it an invaluable tool for neuroscience research. Scientists could test hypotheses about different visual strategies directly in hardware.
Wow, like a hardware model of different eyes.
Potentially, yes. Beyond pure research, it unlocks a new paradigm for application-specific vision. A robot designed for slow, precise manipulation in a factory might need extremely high spatial sensitivity to subtle defects,
Right.
while an autonomous drone operating at high speed would prioritize robust motion detection over fine detail. This reconfigurability allows a single hardware platform to adapt to wildly different perceptual needs, dramatically broadening its potential impact across countless domains.
That's astonishing, a chameleon of vision indeed. Now, traditional analog retinas often face a significant engineering challenge. To integrate more complex circuits, you need more transistors, which typically occupy more silicon area per pixel. This can reduce the fill factor.
Right, the percentage of a pixel's area that is actually sensitive to light.
And that can ultimately impact image quality, right? Less light-sensitive area per pixel. How do the researchers address this common hurdle?
That's a very perceptive point, and it's a long-standing challenge in neuromorphic vision sensors. This deep dive points to a crucial and increasingly viable solution, the stacked chip method.
Stacked chips. Like pancakes.
Yeah, sort of. Imagine this. The photodiodes, which are the components that actually convert light into electrical signals, are placed on one silicon chip, or wafer layer. This allows them to occupy nearly 100% of the pixel area, maximizing light capture.
Okay, light capture on top.
Then, all the complex retinal computing circuits, the OPL, bipolar, and ganglion layers we've discussed, are placed on a separate second chip layer, stacked directly underneath the photodiode layer.
And how do they talk to each other?
They're intricately connected with dense vertical interconnections. These are often called through-silicon vias or TSVs. Think of them as tiny electrical wires running vertically between the stacked layers.
Ah, okay.
This innovative approach allows for more complex retina models to be implemented, packing in far more processing power per pixel on the bottom layer, while simultaneously keeping a near 100% fill factor on the top photodiode layer.
Best of both worlds.
It's a game changer, potentially. It overcomes that fundamental trade-off between circuit complexity and image quality, paving the way for high-resolution, highly intelligent artificial retinas in the future.
When we look at how this particular work compares to other state-of-the-art silicon retinas out there, what truly makes it stand out? What's its unique comparative edge based on the paper's own analysis?
Looking at the larger landscape, the comparison table included in the research paper really highlights several undeniable advantages that solidify this retina's position at the cutting edge.
Such as?
Firstly, it uniquely possesses both luminance adaptation and contrast gain control. Many existing designs might achieve one or the other, but often not this critical biological combination.
Which is needed for real-world light changes.
Absolutely crucial for processing diverse visual inputs across the widely varying light conditions of the real world. Secondly, its utilization of tonic and phasic cells exceeds current standards.
So it sees different types of changes better.
Right. It's not just detecting changes, but differentiating types of change. This enables it to detect a remarkable range of stimuli, from low contrast, slowly moving objects using its tonic-like mode, to high contrast, rapidly changing objects using its phasic-like mode. This versatility is presented as a key advantage.
Anything else?
Furthermore, the paper suggests it achieves this comprehensive functionality with a minimum number of transistors compared to some other advanced spatial contrast retinas, hinting at potential efficiency gains in area and maybe power. And of course, that key advantage of reconfigurability means it's not a fixed solution, but an adaptable, programmable vision platform.
So after all this exploration of its intricate mechanisms and unique capabilities, what does this all mean for you and the world around us? Where can we realistically expect to see this high-performance silicon retina making a significant impact in the near future, or maybe the medium term?
Well, this high-performance silicon retina, or technologies directly derived from it, have the potential to become a foundational technology across numerous applications.
Like what? Give us some examples.
Firstly, think about retinal prostheses, devices designed to restore vision for those with certain forms of blindness, like retinitis pigmentosa or macular degeneration. Current prosthetics often provide very basic light detection, maybe simple shapes. This kind of retina could offer a much more nuanced and adaptive form of sight, better mimicking the natural processing of the eye.
Leading to better vision for patients.
Potentially significantly improved quality of life, yes. Secondly, for autonomous vehicles, this technology could be transformative. Instead of just processing frames, vehicles could truly see their surroundings more like a human driver does.
How?
By distinguishing critical details and movements in incredibly complex, rapidly changing environments, from subtle shifts in pedestrian body language to distinguishing reflections on wet roads from real obstacles, all with low latency and high efficiency.
Okay, what else?
Thirdly, it could profoundly improve robots with more nuanced visual perception, allowing them to navigate and interact with the world far more intelligently and safely, whether in manufacturing, logistics, exploration, or even human-robot collaboration in homes or hospitals.
And even consumer devices.
Potentially, yes. Even in mobile devices, imagine smarter object detection, more responsive camera systems that truly understand motion for better video stabilization or action shots, and enhanced augmented reality experiences that seamlessly integrate digital information with a robust, real-time understanding of the physical world.
So the applications are potentially vast, but no cutting-edge research is without its challenges and areas for further refinement. The authors are commendably candid about some of the current limitations of this prototype, aren't they?
That's an important and honest aspect of good research, yes. They acknowledge several current limitations. For instance, the rectangular pixel design used in this specific prototype is unconventional in standard image sensors, which are usually square, and leads to different spatial resolutions along the X and Y axis. That could make isotropic or directionally uniform vision challenging.
What else?
Also, the large pixel dimensions in this initial prototype, while necessary perhaps to fit in the complex circuitry for a single pixel test, inherently limit the overall resolution that can be achieved in a large array. You can't pack as many big pixels into the same area.
Right, lower resolution overall.
And as you scale these complex analog circuits into larger pixel arrays, you inevitably run into issues with significant parasitics.
Parasitics, unwanted electrical effects.
Exactly. Unwanted capacitance and resistance from the long interconnection wires needed to connect everything in a large array. These parasitics can slow signals down, cause crosstalk between pixels, and lead to increased dynamic power consumption.
So scaling up has its own hurdles.
Absolutely. But they've clearly outlined their path forward in the paper. They plan to address these challenges in future versions. They mentioned focusing on improving component matching between pixels, implementing robust offset cancellation techniques to handle variations, mitigating those parasitic components through clever circuit design and layout, using proper layout techniques like guard rings to isolate pixels and reduce interference, and incorporating calibration techniques to ensure uniformity across a large array.
So, while there are hurdles to scale, they're clearly on a well-defined path to refine and potentially commercialize this truly promising technology.
That seems to be the clear intention. Yes, the groundwork is exceptionally strong.
We've journeyed through the intricate and elegant dance between biology and engineering, exploring how the human retina's remarkable capabilities, its event-driven efficiency, its inherent ability to adapt to extreme light changes, and its specialized cells for detecting different kinds of stimuli are being meticulously replicated and even enhanced in silicon. This isn't just about building slightly better cameras, is it?
Not at all. It's really about crafting a new paradigm for artificial vision, one that can learn, adapt, and perceive with an unprecedented level of intelligence, moving from passive data capture towards active, almost intuitive understanding.
The development of the Tossell-based analog silicon retina represents a truly significant step towards achieving that goal of bioplausible artificial vision.
It absolutely does. It allows us not only to understand the brain's own exquisite computing principles better, just by trying to replicate them, but also to directly apply them to build novel intelligent systems. Systems that can interact with the visual world in profoundly new ways.
Imagine a future where visual prosthetics grant not just rudimentary sight, but truly nuanced perception. The ability to recognize expressions, gauge distances, track motion as effortlessly as a healthy eye.
Or where autonomous systems process visual information with the same astonishing energy efficiency and adaptability as a living organism. Think of the power savings, the speed improvements.
So here's the final provocative thought. What could this mean for bridging the fundamental gap between biological and artificial intelligence? And how might our very definition of seeing evolve when machines truly learn to perceive the world in such a biologically inspired, dynamic, and adaptive way?
It's a fascinating question. Does seeing require consciousness, or is sophisticated adaptive processing enough? This technology pushes us to think about that. It hints at a future where our technology not only mimics, but perhaps even deepens our understanding of the marvels of life itself.
Food for thought indeed as we continue to unlock the secrets of perception, one bio-inspired chip at a time. Thanks for joining us on this deep dive.
My pleasure.
We'll see you next time.